Good morning, everybody. It's a pleasure to be here, and I want to thank, in particular, the conference organizers, Tal and Larry, for this very timely opportunity. Every day, thousands of patients are now benefiting from therapies that target specific mutations on their tumors, and these patients are enjoying months and sometimes years of high-quality life. But today's first-generation targeted therapies are by no means panaceas. They cost typically hundreds of millions of dollars to develop over many years, which means the cost to patients and society is very expensive. And unfortunately, most patients on a monotherapy recur, which suggests a future in which cancer is treated like HIV with a cocktail of these targeted therapies. This is a theme that you'll hear increasingly throughout this conference. Fortunately, we're at a unique moment of time where we have enough knowledge about the way cancer works in the level of an individual cell and ways that one might interdict it with a cocktail. And there's enough drugs in the pipeline that we might have a fighting chance of being able to concoct some of these cocktails for an individual. And finally, the technology, excitingly, is now at a price point and the time for doing the tests that this stuff becomes clinically feasible at a $1,000 genome to be able to test a patient, find out what the possible cocktail might be, and then attempt to treat them. Unfortunately, very few patients have had the opportunity to partake of what I'd call precision oncology 3.0. Some of this is because of the innate conservatism of the medical community and what some might call the misplaced oath of do no harm, because if you do no harm, often you can't do any good. There's just not enough knowledge to satisfy these doctors that we know enough about cancer to actually give it a try. But much more significant probably are the structural impediments that make it very, very difficult to share data and knowledge across institutions, to be able to get access to the drugs that you need to act on that knowledge, to be able to get reimbursement, to be able to conduct FDA-approved experiments with combinations of investigational drugs, even when the patients are very late stage and have exhausted all other options. So these are the issues that we'll be talking about on the panel today. We'll first address what's possible in precision oncology today and in the near future with representatives from 3 organizations that are really on the forefront of this. To add focus and some urgency to the conversation, we're very thankful that Neil Schiffman, a stage 4 lung cancer patient who is actively being treated with targeted therapy, has agreed to let us talk about his case. And Neil, I sincerely hope that if and when the time comes when you need a plan B, that you'll learn something today that will be helpful. And after that, the panel will talk about what we can do as a community-wide action on some of these structural problems. And we're going to end with a call for action in which I'm going to invite all of you to join with the conference organizers, the leaders, the sponsors of this conference to make something happen. So without further ado, it's my great privilege to introduce Carolyn, who will take over the panel.
Well, I want to introduce you to the rest of the panel right now. And Marty, I should have mentioned, for folks who don't know you, that you are a metastatic melanoma survivor. And Marty is that type of guy who, got the diagnosis and then immediately went to work figuring out what he was going to do about it. And since then, since he's healthy now, he's really turned his passion to other patients. And Marty, thanks for all you do. Seated next to Marty is George Demetri, MD, Professor, Department of Medicine, Harvard Medical School, Director of the Center for Sarcoma and Bone Oncology at Dana-Farber Medical Center. Thank you for traveling here. And then we have right next to Dr. Demetri, Dr. Vincent Miller, who is Chief Medical Officer of Foundation Medicine, a molecular information company focused on changing the way patients with cancer are treated and really looking at these personalized medical solutions. And Marty referenced Neil Schiffman, lung cancer patient, non-smoker, triathlete, marathon athlete, cyclist, just an incredibly active, healthy individual until 2011, when he was diagnosed with NSCLC adenocarcinoma. And seated next to me is Ed Staren, MD, PhD, MBA, President and CEO of Medicine and Science at Cancer Treatment Centers of America, a sarcoma survivor, and a real underachiever, it sounds like, too. Thank you so much for all being here. Neil, we wanted to start with you. Walk us through, just briefly, your situation, your diagnosis, and your search for a cure.
Well, I had a good run for 63 years, and then 2.5 years ago, I started a little cough. Didn't think much about it. Little cough became a little bit bigger cough. Decided it was time to see my doctor. And fortunately, I'm one of my gender who has been seeing his doctor for the last 30 years, so he wasn't a stranger to me. He knew all about me. I knew my numbers. In a short period of time, he thought it was necessary. Let's take an X-ray. X-ray led to a CT, showed me the CT, and even I could see there was something different on one side versus the other. And because he happened to be a pulmonologist and was up on his work, he immediately contacted an oncologist for me to see. And between the both of them, they decided that a biopsy was necessary to have. I would have thought that would have been the standard treatment, but I learned very soon thereafter that in many areas that's still the exception for a lot of different reasons because of the invasive processes, et cetera. So the biopsy was taken, and I did a little bit of research and study and found out that there were certain cancers, if you have to have lung cancer, there's certain ones that are better than others. So I started to cheer on myself that I had one of those. And I looked at the numbers to see what the possibilities were. I'm a numbers person, so I played the odds on it. I got the biopsy back in a couple of weeks, it seemed like forever, and that showed that I had the EGFR positive mutation. So that was one of the ones to have. So right now, that's a good one if you have to have cancer, if you have to have lung cancer. So that led me to see a thoracic oncologist because I wanted a little higher level of expertise in my case. So I went nearby here to Stanford, and very quickly, the doctor looked at it, listened to my story, decided that a targeted medication from Genentech, Tarceva, was the appropriate treatment for me. And it was, absolutely. I had a great response to it. It worked for the better part of 13 months. I realized that it will not work forever. So that was my first entry to personalized medicine. From there, I left the personalized medicine stage and I went to the IV, chemo, everybody's the same type bullpen, and was on that for 5 or 6 months. But all during that time, I did my research, and I was looking for clinical trials because I wanted to know what's next out there, what's possible for me, because yes, the pie is everybody with lung cancer, and you cut it down and make it a little bit smaller for adenocarcinoma and EGFR mutation, but I want the little sliver that's me, and maybe there's something out there to help me. So I found a clinical trial that I thought was appropriate, discussed it with my doctor. I hit the trifecta on it because it happened to be one specifically for my mutation. It happened to be one at Stanford, and it was one that she was in charge of. So the best of all worlds. That doesn't happen too many times. And I had another biopsy done, and that biopsy showed that, yes, indeed, the EGFR had mutated to something else, T790M. 50% of the people I learned who have EGFR positive mutation goes to that. And that's what this trial was to discern, whether it would be efficacious or not. 2013 is when I did my year on the trial. And I have to say, unfortunately, a few weeks ago, I had some progression, which I always thought progress was a good thing.
Yeah.
I hear you talk about it in medicine. You want to make progress. It's not such a great thing. And there would not be any more really clinical reason for me to have it. So I had to leave the clinical trial, and I had a good year on it. So now I decided to go back to the Tarceva, my first medication, as a bridge towards the next part of my journey into personalized medicine, some other trials that I am looking at, which I think may be appropriate, and I hope my doctor will also think are appropriate for me.
Now, we have obviously a number of very talented doctors on our panel too who've had a chance to look over your case briefly, but can you each speak to how your institutions would handle a case like this, or perhaps more broadly for other patients who are struggling with this, because mutation is quite common in this battle? Would you like to start?
Delighted to, thank you. Certainly we would, at our organization, have done a small panel of selected alterations to identify which you had. Turns out that with the EGFR mutation and the medication you received, it does work regardless of the alteration, whether you have it or not, but it works far better in those circumstances where you have the mutation. And you're correct, one of the reasons to be able to do that panel is not just to be able to identify a specific approach, but also to identify those approaches that would not be useful. For example, a KRAS mutation, in which case you would not be expected to benefit with that same therapy, and there would be an alternative approach. So we would definitely have done that as well. We probably would have added additional medications, but that's part of the gray zone with which we find ourselves. Certainly the chemotherapy and the targeted therapies. You ended up having, as I recall, you had carboplatin and pemetrexed. There's a lot of debate as to whether or not you may add additional therapies to that. But the good news is, in the circumstance that you have, and to the point that you made, there are options that continue to be available because of precision medicine. Those options include the additions of different chemotherapies. They include the additions of different targeted therapies. My background is in immunobiology, and now an exciting development which fits right underneath that personalized medicine umbrella of various immunotherapies which have been shown to be remarkably effective in this world.
Dr. Miller.
Yeah, so I'm going to wear my oncologist hat and my foundation medicine hat simultaneously. I loved, George, I have to ask you for your slides, because we do need next generation clinical trials, and there have been a lot of efforts to fix that. But really, I think one of the challenges, and sure, you've had a good good case for a lung cancer patient, but that's not much consolation to you. And so one needs to be able to be much more nimble, characterize the tumor in real time, and play it like a chess game where you don't think about move 6 at move 5 with 30 seconds on the clock. You're thinking about it at move 1 or 2, or if you're really smart, maybe, and you know your opponent, maybe even before you start. So I think, and so from my foundation medicine perspective, what we'd like to do in a situation like this is characterize the genomic alterations that are present in the most recent tumor specimen. You've had a lot of therapies that change that. So we've seen T790M disappear under the absence of Tarceva's selective pressure and the presence of cytotoxic chemotherapy. And for now, you've been back on Tarceva, so one doesn't know the exact composition of your tumor, and certainly characterizing the coexistent genomic alterations. And there have been a number described in this setting, including things related to HER2, HER3, EGFR, MET, IGFR, just a very large number of genes and different types of alterations within those genes would really be paramount. But then suppose we find something, what do we do? How do we link you to the trial? How do we make sure that the fact you went back on Tarceva doesn't exclude you? How do we make sure that if your creatinine is 1.3, you can still get that drug, or your squamous cell skin cancer from 27 years ago doesn't exclude you? There's a lot more nimbleness needed in trials now. These are antiquated, generally, system that's been outstripped by the pace of genomic advances. And I think there's a lot of initiatives, both among some of the savvier pharmas and biopharmas at the national level, and with selected companies working in molecular information to try to accelerate and facilitate that process so you can get where we think you need to go, playing the odds again, so to speak.
And Dr. Demetri.
So when we were putting this panel together, I had warned Carolyn and Neil that I wanted to be sure we don't try to practice medicine in front of 1,000 people, that we want to be sure that we're using this as an example. And to me, this is an example of where we as a community have to both be careful, but also look towards the future. And when I say careful, I think we are at risk potentially of hurting the legitimacy of everything we heard this morning by oversimplifying cancer. If you haven't read the book, The Truth in Small Doses, everybody should go out and buy it, because in many ways it talks about how the single driver of cancer, be it BCR-ABL in leukemia, KIT in GIST, EGFR mutations in lung cancer, that is going to be the unusual situation. That is really the unusual situation. Most cancers are far more complicated than that. Most cancers will have a genomic element, an epigenetic element, and probably 5 others that we haven't even thought of. So the complexity is there. But you got to start somewhere. Heck, we've done an awful lot with EGF receptor mutations, before that with the ability to target the mutant KIT in this weird sarcoma. And I think where our community is starting with the genomic complexity, understanding that. T790M, if you don't know about it, what's the problem with that thing is that it basically throws an elbow out and the drug can't get in anymore. So that's not hard. Then you just go back to the structural biology. If you're trying to go after the EGFR, you just find something that slips in underneath that or find a different way of targeting the driver. That's exactly what we've done in other diseases where you know what's driving the cancer. The key question is there something else on top of that? And how do we get away from what George and Frank McCormick up at UCSF have called molecular whack-a-mole? How do we get away from hitting one thing, another thing comes up, and how do we understand when that's even happening? The melanoma case is a great example. That's totally different than the EGF receptor. The EGF receptor gets away from Tarceva by mutating. You get away in melanoma not by mutating the BRAF, but by having a totally different mechanism come in. So you need different strategies in cancer. So what I told Neil is that there are exciting other drugs that are aimed at his problem, because his problem's very clear. It's like structural biology. You go in after something that slips under that gatekeeper and shuts off the EGFR. And there are some drugs that do that. The issue here is back to the issue of how do we design clinical trials. In some ways, he's the perfect person, but many of these second-generation, third-generation drugs limit people who have been on a prior second-generation trial. So there are these interesting and heartbreaking, heart-wrenching issues of access to clinical trial drugs while they're in testing.
Yes, go ahead, Ed.
I might follow. I think the points there were just so terrific because they address the perfect storm that we have ongoing today. I'm a sarcoma survivor. 10 years ago, it was a discussion whether or not there was going to be any role for any adjuvant chemotherapy, and the data was dismal. And in just that short 10-year period of time, the information from whole genome sequencing and the various potential opportunities there are mind-boggling. We have a role, it seems to me, and this is a perfect storm audience of multiple constituents that end up representing all the different facets of patients, industry, the providers, payers, government, etc. You made the point that we will have increased information, and therefore there might be increased options, at least theoretically. But without that cooperative advocacy of all of those parts working together, nothing will happen. I admire Neil so much, as I know as physicians we all do. To have a patient come up there and bare their soul to help advance this cause is the greatest advocacy that we can have. And it turns out that those hopeful options, our responsibility is to be able to ensure that we empower patients like Neil and make sure that we collaborate in our advocacy role to ensure that that access occurs once you end up having these new and exciting opportunities and therapies available.
Marty, I think this is a great opportunity for you to step in and talk a little bit more about what you are trying to do.
Well, I started Cancer Commons out of personal necessity. 15 years ago, I was diagnosed with metastatic melanoma, which at the time there were no effective treatments or cures. And I was given 12 months median survival, and I was freaked out, like all cancer patients, numb, paralyzed. And what made it worse was going to many experts, and I had the means. I went out and I probably talked to 24 people who were at the cutting edge of the field, and everyone told me prognosis isn't good, but here's what you should try. And most of them were different, and there was no data to make any decision. And I knew as an internet entrepreneur, computer scientist, cancer survivor, that if I got my health back, that I was going to do something about this. And so that led me to create an organization which is part clearinghouse, part catalyst, part convener, to bring all of the stakeholders together so that we could, in fact, learn from every patient. The basic goal is to build a consensus knowledge base of what are the molecular subtypes of different cancers and what is thought to be the best way to treat them or the most appropriate trials. And then to ask make this information available to patients and physicians and anyone else who wants it. Let patients use that to guide their treatment or get other ideas because it's a learning system. And then, most importantly, to report back what their experiences were, what outcomes, what responses they had, so that the community as a whole can learn. And in order for that simple learning loop to happen, we need cooperation from so many different players that are today reluctant to share any data, usually using patient privacy as an excuse and to hold on to data that they would like to regard as proprietary. I'm really pleased to say that this message is catching on, and one group that it's caught on with big time is everyone who's up here on the panel. That was one of the conditions for getting them up here on the panel. So I'm hoping many more of you will join us in this effort.
And you talked a little bit about how do you learn from patients and their experiences with this. Neil, I know you've been very proactive in speaking with every step of the way. You've gone back to Genentech and told them what needs to be different, perhaps with their trial. You've spoken with the folks at CyberKnife about what they can do differently. I don't know if all patients are as involved as you are. And when you have people who come to your institutions and then return to the community, how do you track them? Are you able to keep track of those results?
Part of it is maintaining contact. We have what we call cancer fighters in our organization, and it turns out that you don't have to seek those individuals out. They want to be empowered. They want information that they understand. They want it translated into terms that they can practice, and certainly they want to be respected so that you identify what's valued. But they have a big story to tell, and what we're dealing with is this whole concept of N of 1. It's not 1,000 patients that need to get a message across. When you end up having no options and you now have a new option that makes an enormous difference, that shows tremendous response, the whole mindset needs to change. And those individuals do that, one patient at a time. We need to be working in advocacy with the government. Certainly, Rich Pazder and the FDA has come forward with all kinds of innovations and approaches to be able to enhance availability of drugs. We need to be working together in terms of the IT environment. There's tremendous challenges there, from de-identified data to access to that data, interoperability, certainly secure information, but information that's flexible, the ability to be able to have standards with data. The problem is that we are not collaborating at a level that we need to. We're not cooperating at a level that we need to. We all know the problem, but without that collaboration, we won't go forward, and patients are going to drive it. I'll finish with one last statement, but I think it's a great example. I heard the saying of approaching individuals in the Titanic that we haven't yet tested the lifeboats. These are individuals whose lives are in peril. They really don't care that you haven't tested the lifeboats yet.
Yes.
Carolyn, I appreciate what Dr. Staren was saying. I think it's important, this audience probably knows this, but it's important to reemphasize the fact that we have very peculiar incentives in American medicine. Ralph Snyderman brought this up before, and I think unfortunately they've continued. I'm really hopeful that the ACOs will change that and we look to a different system of incentives that will hopefully provide better care and actually incentivize a rapid learning community in a different way than the fragmentation of care that we see today. More importantly, what I want to emphasize is that less than 10%, far less than 10% of patients with cancer in this country participate in clinical trials or clinical research. What happens to the other 95% is completely lost information, absolutely lost. We might barely get it from tumor registries about incidence, that people actually get a cancer and then when either they live or they die. That level of 35,000 feet up data is what we even now have trouble tracking, let alone trying to say there is a new drug that hits BRAF mutant patients, and then rapidly trying to say, How does it work if somebody's got a BRAF mutation? Because this is the other challenge. Most people in the room know this, that if you have a BRAF mutation in melanoma, that those drugs work like a charm, even if only for a year or so. But if you have a BRAF, the very same BRAF mutation in colorectal cancer, it's essentially useless. So there's a context of vulnerability issue here that goes beyond the simple annotation of the gene problem. It goes into other pathways that we still don't understand. So how many such negative N of 1 experiments does the community go through before people finally get it? When vemurafenib was approved, I can guarantee you there were a bunch of patients who almost certainly got that drug, and we knew that it wouldn't work. We already knew that from a small clinical trial. But I think as a community, as a society, as a country, we have to get better at this and not lose all that information out there. That's what appeals to me about Marty's donate your data idea.
Yeah, I just wanted to say that the concept that people are alluding to is what I call global cumulative treatment analysis, where every patient is a trial of one and we aggregate the learnings, not just the data, but what's learned from every patient and make that available in a timely fashion so that it can benefit the very next patient coming along. There was a paper that we published in Nature Reviews just last Tuesday called Rapid Learning for Precision Oncology, and I would be happy to send all of you who don't subscribe to Nature Reviews a PDF. Just send me email.
And Vincent, I know you wanted to say something.
Well, the first thing was the GCTA. Was that a pun? Or is
Yes,
that an appropriate accurate?
it's a pun.
Okay, very
Explain why
no
it's a pun.
surprise. That's great. That's great. So, I guess this is similar to George's thought, though. Is one, of course, the predilection of scientists to publish their positive results and not their negative results. So there's probably innumerable questions we could answer if we pool together even a significant subset of these 95% of patients who don't go on trials. And as a corollary to that, I think many of us feel that the low-hanging fruit of cancer genomics and matching a kinase inhibitor with a mutation, we've probably found the vast majority of those. That cancer biology is far, far more complicated. And many of our best hints, if we're going to condense those timelines, may result from this interplay between genotype and phenotype, because things are going to fall out that are completely unexpected, paradoxical responses to a drug that one would think would never work in a certain situation, or the converse, unfortunately. So I think that's how we think about it, both oncologically and in foundation medicine. That if you have the anchor of the rigorous genomics, as George said, we got to start somewhere. That's not the total solution, but when coupled with this other piece that Marty makes such an impassioned plea for is a way to make it relevant in your lifetimes.
And let me just make one other point about that, because in addition, we heard before that we're turning cancer into a chronic disease. We've been hearing that for a decade, and it is true. But we're also turning cancer into a rare disease. I used to love it when the NCI would say, cancer is at least 50 different diseases. Well, heck, I take care of GIST patients. That's 1% of all cancer, and in GIST, there's 50 different kinds. So cancer is thousands of different diseases. We are turning rapidly cancer into a panoply of thousands of different genetically, genomically, et cetera, defined problems. And they're going to need a much different way of analyzing how to make sense of that, and I think that's what we're starting to do.
Well, let's talk about the challenge of changing clinical trials as we know them. I mean, that is a huge nut to crack, isn't it?
It is, but it's not one that's not receptive. I end up being glass half full, and I do believe that the problem is recognized, and we have terrific leadership that will address that concern. That entails a different mindset on how we run clinical trials that we grew up with. No longer is a 1,000-patient over 10-year type of a mentality appropriate. We certainly need to change our statistical analysis. The statistics that are utilized for standard clinical trials probably don't apply to the N of 1 type of mentality, whether we move into Bayesian type statistics or what. That is a different kind of addressing the need. And we will, I believe, evolve along those lines.
Did you want to speak, Neil?
Yeah, I did. About clinical trials and just personalized medicine in particular. So this conference, personalized medicine, and I looked at the program, and I finally found one patient on the program, which I thought was just a little bit weird if the focus is supposed to be on patients. So there should be more part of it in some way. Some of us got the patient advocacy gene and had it turned on, so we actively got involved. My pathologist missed that when he did the EGFR, and we looked at it again, and then he found it. He said, that's why you're the way you are. So that made me contact Genentech and Accuray and talk to people there, and they were shocked because they never hear from patients, partially in some cases because they don't ask patients about their treatments and how it went and how can we make it better. And when it comes to the clinical trial that you mentioned, so I was on a clinical trial last year, and it was a great year for me. And after being on it for 4 or 5 months, the gene must have hit me again a little bit more because I thought it was important that Clovis Oncology, the company running the trial, to whom I was only a number in a cohort, they should hear from me because they have to know what I'm thinking about their trial and how it can be better, not only for me, but for maybe all of the other patients that they have on the trial or in the future. So when I talked to a couple of people, they said, well, that's not really done. I said, don't tell me that. That only makes me want to do it more, of course, like all of us. So I contacted the company, and someone contacted me back a couple of weeks later, and we got approvals from the FDA, from Stanford Ethics Board, and I went in and talked with them along with my wife and had a great session. I think they heard from me as a patient things that they would not have thought about, and I learned things from them that helped me with my trial, and then also that I could pass on to other people on the trial. So I hear a lot of times, let's make the patient involved. It's like, you can't wait for the patient to come to you. Most of us are pretty sick. Most of us haven't gotten that patient gene activated, so they're going to go out to you. You have to reach out. You have to ask the question, what can we do to make the experience better for you? Okay, how can we help you and help somebody else? How can we help you be a mentor to the next person who then becomes a mentor to 2 other people, and eventually the N of 1 maybe becomes N of 3, of 5, of 10, etc.
Really quickly, can I just get a show of hands? How many patients are with us today? I know this was the first time the conference was really opened up to patients, and have some of you. Terrific. Welcome. Thank you for coming. I'm sure this has been very informative for you.
Can I ask a question?
Yes.
How many, since we have a lot of industry leaders here, how many of the leadership in this room have patients in your board meetings or those type of events? When I joined CTCA, it was a harrowing experience to find that we never start a board meeting without listening to a patient. And I can tell you as a CEO, plenty of times there were things that came out at those meetings that were a little embarrassing. But the fact of the matter is that is our customer. And so for those that have not done it, I would encourage you in the strongest possible way to think about having patients at those type of meetings and hear from them so that you can address their issues firsthand.
Marty.
Yeah, I just wanted to put a capstone on the discussion about trials, which is that if you take what George said, which is that there's probably an unlimited number of varieties of cancer, and you take that in the context of saying these cancers are going to have to be treated, most of them with some combination of drugs, then, and there's hundreds of these drugs that are in development, you just think about the combinatorics of it, right? There's no possible way that you can run these large trials or even small trials and test all of the rational possibilities of drugs that make sense for all of the patients that are out there. So that means that we have to really fundamentally rethink what we're doing. And I think that the best way to approach this, not for all patients, but for the patients who really are off the map in terms of standard of care, is to be able to deeply analyze, reverse engineer their tumor. And this is going to take a lot more data than anyone is really thinking about today. We're talking about perhaps a terabyte of data on an individual patient based on all of the different panomics that one can measure, not just genomics, but expression and epigenomics, metabolomics, et cetera, and use that together with network analysis, sophisticated ways of linking up all of the alterations in order to understand at the level of pathways what's going on in this tumor and how we might interdict it. And when I talk about then using drugs to treat those patients, this is something that's done not over years, but immediately on one patient. And the FDA doesn't seem to have a problem with this because you're not putting a lot of people at risk. You're asking for compassionate use of drugs. The key problem is getting the drugs out of the drug companies to be able to test these things. But once we have them, then the drugs become probes. You're not just treating the patient, but every time you treat a patient in a rational way, you're learning something about the disease and about the drug. And by pooling learning at that level, we're going to make progress a hell of a lot faster than we've ever done before.
And let me add that I think we have made tremendous advances even in our clinical trials. The 80s, when I trained as a medical oncologist, was the era of toxic Scrabble. You know, give your drug a name. Adriamycin was great because it could be a vowel or a consonant. So depending on what acronym you wanted, you could just put adriamycin in and come up with a clever acronym. That was pathetic. And now we have mechanistic reasons to use a drug. You know you've got that pathway turned on, you know you've got a mutation. So the two advances that I think we've seen in clinical trials based on mechanism really have to do with that. The mechanism-based basket study. It was awful when Gleevec was first approved and Novartis was giving it away like water. I was saying, what have you learned? They say, well, nothing. We give it away and we never hear anything back. I said, that is pathetic. You know, let's design a rational basket. If we have a disease where a target of the drug is turned on or likely to be there, let's design a study and let's learn something from it. And that study led to 5 different FDA approvals for weird diseases, a weird kind of sarcoma, a weird kind of blood cancer, a couple of other things, on a single day in 2006. So they were very happy about that, and that was one of the molecular basket studies. And now molecular basket studies are all over the place. Anybody who's got a targeted therapy can say, if you've got this mutation, you get into a basket. The other one are the exceptional responders. And exceptional responders could be either exceptionally good or exceptionally bad. You learn a lot from studying resistance. So those things, both mechanistic and structural and genomic, teach us rapidly where a drug works, where it's not going to work, and then we can go back and advise on the next generation of trials, advise our colleagues in the community, it's not going to work, get people like Neil to participate in the next generation trials faster, and frankly accelerate the whole drug development process. It is pathetic how long it takes in general. But I think with this kind of science and with this kind of collaboration and openness, we can move much faster.
And how do we encourage the collaboration, both from the patients? I know you have the gene where you want to get out there and share, but from the patient's perspective and also from the doctors, from ordering those standardized gene panels or moving forward in that way.
Well, let's just take Harvard. Harvard is famous for not collaborating even within itself. So we have the biggest comprehensive cancer center in the country, a consortium cancer center, and we essentially have a social experiment going on where on one side of town, genomic tests are being sold as a standard. On the other side of town, we're doing it as a research project funded by philanthropic trustee funds. And we view this as research. And I think some of this is when you give people something, incentivize them to play, everybody wants to do right by patients. But as, again, Ralph said, sometimes the incentives are perverse. So we have to pay attention to the incentives and make the research incentives and the clinical incentives and the patient incentives all align. Very often we don't do that. Academic medicine, in many ways, has all these subtextual incentives that really prevent collaboration.
Well, to that point, we actually have varieties of disincentives. I heard that there's availabilities of compassionate use. Compassionate use is not easy. There are plenty of hospitals that don't have IRBs, and that's a requirement in your first step. The IND forms for the physician are not easy to fill out. And then there's the question of cost and payment on that particular drug. You get into the standard clinical trials, and even some of the areas of clinical trials, if we do adjust the clinical trials and we end up having smaller ends and we do it for a shorter period of time, we still have a responsibility to follow those patients for a longer period of time, even when they get into market because of safety. So those kinds of decisions need to be made up front so that we can have that collaborative effort. We need to agree as a group on the rules of the road.
Vincent?
Just add, Carolyn, that the other challenge / opportunity is the vast majority of oncologic care is administered in a community setting, 80, 85, 90%. It's not where Neil's fortunate enough to be treated. It's not where George worked. It's not where I worked. It's not the patients, Ed would see. So there is not only a huge educational gap, a couple of logs away from what the talks you're hearing about today as far as understanding, but these guys are just facing a boulder that they're trying to push uphill to get a patient in any sort of study. So there are some pharma initiatives, without mentioning any particular pharma, of bringing trials to the doc in the community setting if they have a molecular match or hit. And that gets back to this issue around the nimbleness of the trial. We must make it easy to do and have a stake in the game, not the doc sending the patient off who they've known for 3 years with their CAT scan and their PET scan and their bone scan and their pathology to be seen at a center of excellence, only to come back 3 weeks later there wasn't an opening on the study.
Yeah,
That's true.
that's got to be a huge challenge. Marty.
I just wanted to give a specific example of a straw man of the kind of alignment that people are talking about. If you think about a patient being treated outside of a study, outside of a trial, and there's a strong rational basis that playing chess with the cancer, that there's that some tests revealed a set of alterations that we could hit with a combination of drugs. An ideal in my mind would be that with that strong evidence, drug companies should be eager to make these investigational drugs available for experiments, not in 1,000 people, but in that one patient. And if at no cost, and if the drug works, then they ought to be entitled to get their costs reimbursed, or if it's an approved drug, to actually get paid for the drug by the health plans, the insurers, because the drug works. If it works, otherwise so the contingent reimbursement, which is much more common in Europe, would be something that would work here as well. And the final thing that would align interests is that if the FDA would be more liberal at allowing combinations of drugs to be tested when there was a strong rational basis for them and no prima facie evidence that they were going to be mutually toxic. In one patient who was dying anyway, I don't see any ethical issues with that. So if you do that and you capture the learnings from it, that's the opportunity to accelerate by years, because whenever you talk about a study or a trial that you have to write up a formal proposal for and get approved by an IRB, by your university, by the FDA, by the drug company, raise funding for it, accrue the patients, run the study, publish it, and then wait in escrow for 4 months till ASCO decides to let the results out. You're talking about a decade as a minimum. And think about that as a comparison where some researcher in a lab is working on tumor cell lines from a patient who's dying, has some remarkable success with a combination therapy, and doesn't have any idea who that patient is because it's all de-identified. Think how we could cut a decade out of the system by going and connecting that researcher and that patient who need each other. And that's the way this kind of rapid learning study could work.
I know we have some microphones available in the audience, so for those of you who have comments or questions.